Today's lesson is looking at immunotherapy, which is basically the modification of our immune system to be better equipped to deal with any diseases that emerge in our body, right? So on its own, our immune system is already well equipped, it's pretty good at doing what it does, but there are some flaws or there are some weaknesses that it has that diseases and pathogens exploit. Immunotherapy is basically us humans taking charge, understanding where the weaknesses are, and using different techniques to enhance our immune system to be better equipped to deal with these diseases. All right, so let's just do this question. Zika fever is a rapidly emerging viral disease. Rapidly emerging, what does emerging mean? Who can tell me? Yasmin, what do you think? Emerging versus re-emerging. What is emerging? Okay, let me give you an example of a disease that's currently emerging, right? Monkeypox. Right, I'll give you some details about it. It was first found within laboratory monkeys, and now it is an emerging disease. Why? Something about it that is making it considered as an emerging disease. Any ideas? Sure. It's new to the population, but more, that's one thing, but more importantly, it's emerging in multiple outbreaks, right? There are multiple outbreaks that are outside of its normal location. So let's say something like we have a virus that's endemic to Australia, something like, I don't know, make up a virus in your head, but suddenly it's emerging in different parts of the world, right? There's one in Europe, there's a few in US, there's a few in South Africa. That means that that is an emerging disease because it's crossed its borders, it's emerging in multiple outbreaks, and also, yeah, it's new to the population, right? Those two factors cause it to be emerging. Re-emerging is basically when it went down and then the case numbers are re-emerging back up, right? So it's in the word itself. So make sure you remember the difference. So it is commonly transferred from one person to another by the Aedes mosquito. Zika fever in people were discovered in Uganda. Thought that a bite from the mosquito had transferred the virus from monkeys to humans. So this is an example of what kind of disease. And again, Janita. So the disease is transferred from monkeys to humans. So that makes it a special type of disease. Where do you go to see animals? Zoos. So what type of disease is this? It's like zoon. It goes with an N after that. Zoon. Zoonotic disease, right? Zoonotic diseases are those diseases that cross the species barrier between animals and humans. So this is an example of that. The symptoms of Zika usually are a fever, mild. 80% of infected humans do not show any symptoms. Infection of pregnant women, however, can cause severe defects in their babies. So going to the question, when scientists attempt to identify a particular disease, they can look for a specific antibodies in infected humans. Scientists trying to identify Zika fever infections found that testing for the antibodies produced against the Zika virus often gave them incorrect results. This was because the antibody tests that had been developed could not always identify the difference between antibodies produced against the Zika virus and the antibodies produced against other viruses. Explain why making a correct identification of a viral pathogen is important in the control of a disease. So all this is fodder that meant nothing. It's all about this last part here. Do you know this? So that's going to be sometimes important as well. So try to understand what the question is before you even start reading all this irrelevant information. Because if you had wasted time reading all this, question itself being so straightforward, you would have not noticed it, right? All right. So what are the importances of identifying any pathogens, viral pathogens, in the control of a disease? What's one? Any ideas? So think about any pathogen in general. The reason why we need to correctly identify it There's a few reasons. The first one is what? Right? Can have a mutation. So you need to better describe that. What does that mean though? Why does can have a mutation have anything to do with this? All right. So let's put it into a different context, right? Let's say we're talking about coronavirus here, right? Let's say this is the emergence of coronavirus happening in Wuhan. Why was it very important to quickly identify what is the pathogen, what is the virus itself, and learn everything we can about the virus at that very point when it was emerging in Wuhan? Why would we have needed to know everything we can about the virus or control the spread? Right? It's that straightforward, actually, because specific pathogen methods of transfer, by understanding what the pathogen is, we can also control the spread and also treat it using the right antiviral drugs as well. So there's two points to it. One is that you can understand how to treat the virus or the disease that it causes. Second is that you can control the spread by knowing how it is actually being transferred, right? Is it airborne? Is it direct physical contact? Is it fecal oral transmission? We don't know until we identify the pathogen. So that is one of the first things. The second thing is that they may require specific controls such as antiviral drugs. And incorrect identification can also lead to increased spread and infections. So one, there's always talking about controlling the spread. Two is treating it right. Third is to, if we don't identify it correctly, then those can actually become a problem as well. Yeah. So this is how we would do it, right? Isolate the antigens, and this would enable us to produce vaccines as well. Moving on from that, okay, we'll actually go on to the topic part. So today's lesson, we're looking at immunotherapy. You guys will finally learn what a monoclonal antibody is. I need to stress this very heavily. Monoclonal antibodies will definitely be asked in your SAC and also your preceding examination at the end of the year. So you guys definitely need to know what monoclonal antibodies are. You also look at how we use monoclonal antibodies in treating cancer and any other problems, actually mainly treating cancer, in fact, with monoclonal antibodies. Immunotherapy is the strategy that we use where monoclonal antibodies are made specifically to treat either cancer or other forms of autoimmune diseases that we encounter. So these are all your words that you need to know. Move on to that topic. So immunotherapy in general is a medical treatment that works by modulating the functioning of your immune system to be better equipped to treat other diseases that are above its pay grade kind of thing. So think of cancer, autoimmune diseases. These are much more difficult for our immune system to deal with because, for one thing, cancers and autoimmune diseases are piggybacking on our own cells, right? Especially cancer. Cancer is literally when your own cells have gone haywire and started to reproduce uncontrollably. But since they're your own cells, they also still have your self antigens, right? The antigens that are inherent to all your own cells. So that makes them somewhat unrecognizable to your immune system or less likely to be found by your immune system. So as such, some immunotherapy will be needed to modulate that our immune system to be better equipped to deal with these kind of diseases. And there's two ways that it can primarily change our immune system. One is to induce our immune response, induce or amplify our immune response. Second is to basically prevent or reduce our immune response. So it's either activation immunotherapy or suppression immunotherapy. Either one can happen and can be different depending on the situation. So the 4 types of immunotherapy that you guys are gonna have to remember. So each of these are basically us as humans modifying our immune system in one way or another to be better equipped to deal with the disease. The first one is your dendritic cell therapy. This is basically taking a dendritic cell, which is a antigen presenting cell from our second line of defense, and programming it to be basically targeting a specific tumor by giving it tumor associated antigens, right? So basically take it out of your body, prime it with the antigens that are associated with a tumor, and that by doing that, the antigen presenting cell then is going to specifically target for that tumor that we've primed it with. Next is your CAR T therapy. This is basically a modification of your T cells to recognize and destroy cancer cells. Basically, for this one, rather than giving it an antigen, we are basically programming the cell, the immune cell, to have the receptor for an antigen specific to the cancer cell. Because at this point, this immune cell, the T cell over here, wait, let me get to the picture. All right. So imagine if this is the T cell itself, this is the cancer cell, right? Normally, the T cell would have receptors on the surface of its cell to basically identify which one is a pathogen, which one is a disease, which one is a pathogen, and identify it and correctly target it. But with a cancer cell, it might not have the receptors necessary to identify it. Basically, without the right receptor, it won't even bind to the cell and it won't even recognize that cell. So what this therapy, immunotherapy, is about is equipping the T cell with the right receptors such that it can actually bind to the antigens on the surface of the tumor cell. And when it can bind, it will basically destroy that tumor cell, that cancer cell, same thing. All right. After that is your antibody treatment. This is where most of your monoclonal antibodies come under. And this is the one where you're going to have to put a lot of focus. Basically, this is talking about creating and using antibodies to stimulate and enhance the functioning of the immune system. So here's a little pop quiz for you guys. If you are given a bunch of antibodies, what type of immunity is that? The hospital has prepared a bunch of antibodies for you, and they will inject it into your body for it to do its job. What type of immunity am I talking about here? Remember the 4 categories, right? There's active, passive, there's active, passive, and then there's artificial and natural. What is the type of immunity when there is a creation and injection of antibodies within your own body? So you are given antibodies when they are produced outside of your body. So what type of immunity is that? Can I get? Yeah, put. All right, so let's work through it. All right. Is that a natural or artificial way to get antibodies? What do you think? Is being made by your own body or is it being made outside of your own body? Outside. So is that artificial or natural? Artificial, okay? Sorry, that's not artificial or natural. That's active or passive, whether it's being made inside your body or not. So if it's not being made inside your body, that is passive, right? Next is talking about whether it's natural or artificial. So the fact that this is made basically outside of the human body, it is medically made using medical intervention, that makes it artificial. So this is basically artificial passive immunity, where antibodies are made outside of the body and they are then injected using medical intervention into you. So that's why it is artificial passive immunity, right? Make sure we can identify which type of immunity based on the situation that they give. Next is cytokine therapy. This is basically using immune molecules such as interferons or interleukins to modulate the effect of your immune system. Right. So let's look at the first two before we go into the monoclonal antibodies part. So looking at your dendritic, this is a form of activation immunotherapy, basically enhancing your immune response. And this is activated by basically vaccinating tumor-associated antigens, basically the antigens that are associated with the tumor or cancer cell. So let me try to paint a better picture of this. They find the tumor cell in your body or the cancer cell in your body. They analyze that cell to find the antigens that it has. Once they know the antigens that that tumor has, they inject you with it as a form of vaccination. And through this, your dendritic cells can pick up those antigens and also then be better equipped to fight those tumor cells. This can also happen to the manual infusion of dendritic cells, meaning that rather than just injecting a vaccination into your body and hoping the dendritic cells take hold of that, they basically harvest dendritic cells from the human first, then they expose those tumor antigens to the dendritic cells, then they select the dendritic cells that have actually taken up those antigens and then only inject them back to your body. So this is reintroducing the dendritic cells that have associated themselves with the antigen. And once we reintroduce them, they will basically trigger an immune response and they will specifically target the cancer cells because they were already equipped with the antigens, basically the targeting system for the tumor. And now they can just look for the tumor and find it and destroy it. Right? Any questions about this one? So this is basically equipping the dendritic cell with the target. Basically, what does the target look like? We're telling the dendritic cells, and those dendritic cells then pass on the message to all the immune cells that come after that. All your T cells and B cells that emerge after that will be equipped to deal with that specific tumor cell. All right, moving on from here, we have your CAR T therapy, right? This is also another form of activation immunotherapy where your immune system is being enhanced, right? So for this one, T cells are extracted from the patient and a gene coding for the antigen receptor is added to its DNA. So like I said, right, these T cells do not have the receptor for certain tumors, so they can't even interact with those tumors. So rather than working around that, what they do is that they extract those T cells from the human and then they basically genetically modify that cell to have a antigen receptor specific to the tumor on its surface. So this is a form of genetic modification, and they can use all the different techniques to do that, right? Your CRISPR, your and basically any genetic modification technique you can think of, they can use it here to make the T cell then have the antigen receptor on its surface. And once it's done, once it actually has the ability to produce that receptor, it will insert that receptor into its membrane and thus gain the ability to recognize the cancer cell antigens, right? So I hope everyone understands this straightforwardly, but let me try to shape it in a different way for those who don't. So at this stage here, right, let's say you have a tumor growing in your body. And the reason why that tumor can't be dealt by your immune system, every time your T cell tries to approach that tumor, it doesn't have the right receptors on the surface of the T cell for it to even recognize the cancer cell. So okay, imagine if this is the T cell, it needs to be able to recognize this tumor cell before it can even do anything. But at the surface of this tumor cell, let's just assume this. So this tumor cell only has these type of antigens on its surface. And this T cell doesn't have any receptor that can actually bind to that antigen and recognize the tumor. So what it needs is that this T cell needs a way to have a receptor that binds specifically to that antigen, and your immune system doesn't have a way to actually do this. So what we do, we program your immune system to be able to actually do that. So how we go about doing that? First, we remove T cells from your body. We then insert a gene that is specific to the we insert a gene that can allow it to program for the receptor that is specific to the antigen that the tumor has. So basically a gene for CAR. This is cancer antigen receptor, by the way. That's what CAR stands for, cancer antigen receptor. So the gene for cancer antigen receptor is added and inserted into the T cell. And once the gene has been inserted, obviously the cell will produce that protein. And once the protein is produced, it will be added onto the surface like a machine adding a new part. And once it has gained this new upgrade, it is able to actually recognize the tumor cell. So millions of those CAR T cells, which are basically the modified T cells, which are cancer antigen receptor T cells. Once we produce them, we reproduce them over and over, and then we inject them back into your body. And now a bunch of these CAR T cells that can actually recognize this antigen are in your body now. Yes, sir. Cancer antigen receptor. Oh, chimeric. Chimeric is because of how it's produced, right? It's produced by the cell itself. So that's why it's considered as chimeric. Yeah. Oh, I see. Cancer. Yeah. It's chimeric, sorry. Chimeric, it's made by itself. That's why it's chimeric. If you have never heard of the word chimera before, the ancient mythological creature that's basically made up of a bunch of different animals. So that's why this here, chimeric antigen receptors, these are basically made to be made. Let me find a better way to do it. It's programmed the T cell to make its own antigen receptor by inserting the gene into it. And then once the T cell itself makes that receptor, we will reproduce that T cell over and over until we have an adequate amount. And then we'll inject your body with those T cells. And now, since they have been able to produce that specific receptor, they can actually bind to the antigen that is in your body, the tumor that is in your body, and seek out and destroy those cancer cells. So yeah, this is your final product here, right? So those millions of chimeric antigen receptor T cells are grown and infused into your patient. And once they are infused into the patient, they will actively target for the cancer cells and seek out and destroy them. Any questions here? All right, moving on. And now we're looking at our antibody-based immunotherapy, right? So this is specifically where we use antibodies in line with our immunotherapy strategies. So what are monoclonal antibodies? They are basically lab-made proteins that can be used to treat a number of different diseases. They will bind to specific antigens only. So like you guys know already, antibodies are very specific. They will only bind to those that their antigens are, those antigens that have the specific shape that correlates to the antibody itself. So antibodies are so specific that if they don't have the same shape as the antigen that it wants to bind to, it won't actually bind to it. So make sure we are aware of that. So targets only the cells that have those antigens it's been programmed with. And when it comes to immunotherapy, antibody-based immunotherapy, the main focuses that we use it for is for fighting cancer and autoimmune diseases. Basically, they are best equipped to fight cells with self-antigens. And looking at it, it can also be used in a non-immunotherapeutic way, where it actually doesn't improve or suppress your immune system. It just acts to fight the diseases. So here we are focusing specifically on it activating your immune system. But when we're dealing with monoclonal antibodies, it doesn't always have to activate or suppress your immune system. Sometimes it can just act to fight the disease without changing your immune system at all. It all depends on the situation and what type of treatment is being done here. Oof, you guys can't read that, can you? Sorry about that. Just give me a sec. All right. So when we're looking at monoclonal antibodies, we're going to look at one specific way of producing them. This is a more traditional way to do it. There are definitely more ways to do this now, but we're looking at a traditional way, and your assessment is going to definitely ask in a more traditional sense. Okay, the first zoom in. Let me zoom into the picture and explain from there. One thing at a time. All right. So the first part of remember, these monoclonal antibodies are used to fight off cancer and autoimmune diseases. So before we can actually start using antibodies, we need to know what we're dealing with, right? Identifying the pathogen. As always, that must come first, identify the pathogen. So first thing we do, identify what cancer cells are being sprung in your body, that they are growing in your body, and isolate the antigens that are present on that cancer cell. That is the first step. So identifying the pathogen and getting information from it. Next is that, very weirdly, we need to basically vaccinate a mouse with that the antigens from the cancer. This is specifically to trigger an immune response from the mouse, which will be needed for the third step. So at this stage, after the mouse has been exposed to the antigens, its immune response will be triggered, right? Second line of defense, third line of defense. So we are counting on its third line of defense, where it actually produces B cells, B plasma cells. And we basically those plasma cells from the mouse, right? We want those B plasma cells that are that are specific to those antigens. We want them, so we expose the mouse to that antigens. We allow its immune response to build to make the B cells, the B lymphocytes, and then we extract those B lymphocytes from the mouse as well. Moving from there, we then basically fuse the mouse B cells with a specific type of cell called a myeloma cell to create something known as hybridomas. Okay, this step is done to ensure the survivability of those B cells, because B cells by themselves don't do a good job of surviving outside of the body, right? In vitro, basically, these B cells will die off very quickly outside of the body. So we need a way to be able to keep them outside of the body for prolonged periods of time to allow them time to produce more antibodies as well. So what we do, we merge them with something known as a myeloma cell, which is a cell that can basically reproduce without end. Myelomas are a unique type of cell because they can differentiate forever. As long as they get the right amount of resources, everything else is right, they can actually reproduce forever. In fact, we have certain cancer cells. So myeloma cells are a type of So the reason why these myeloma cells are chosen to fuse with B lymphocytes is because they do not grow well in vitro. In vitro means outside of the body, whereas myeloma cells have the ability to grow indefinitely, and as such, they will produce large quantities of antibodies as well. So that's why we fuse them together so that we can basically increase the outcome of the B cells. Once we've done that, we basically select for the right type of hybridoma. Remember that hybridoma is the name given to the fusion between a myeloma and a B cell. Once we fuse them, we call them a hybridoma. So once we've already fused them, since we've extracted a bunch of different B cells, some of them can be specific to different antigens. We need the one that is specific to the antigen we're trying to isolate. Antibody we're trying to isolate. So we will select for the appropriate hybridoma that actually binds to the antigen that we have. Yeah, basically that's how we know whether it's the right right hybridoma or not. We basically expose it to the antigen that we have, see which one actually binds together with the hybridoma. If it actually binds, that means that is the right B cell, right? That is the one that will produce the right antibodies for the tumor. And once we do that, we basically collect and purify the antibodies, right? So one more time, this time I'll just show the words instead of the pictures. So first thing is isolate an antigen that is present on a desired target cell, essentially the cell causing the disease. And then once we do that, we basically vaccinate an animal with that antigen. And this is meant to trigger an immune response to produce B lymphocytes, basically B plasma cells. Once we do that, we extract that B lymphocyte from the spleen of the mice, which is basically where B plasma cells are going to be found at one point. And then after that, we fuse those B lymphocytes that are going to die off if we don't do anything about it with something known as a myeloma cell, produce something known as hybridomas, which are much longer lasting and also can produce more antibodies. And once we've actually made this bunch of B cells, we isolate the right type of hybridoma that can produce antibodies that are going to bind to the antigens that we actually had at the start. We basically select for the hybridoma that is specific to that antigen. And once we do that, those more antibodies are made and then purified and administered to the patient. All right? So steps are straightforward if you follow along. Basically, let me actually give more meaning to it. Antigen A is isolated from the tumor cell in your body. Then that antigen A is injected into the mice to trigger an immune response. The mice will produce a bunch of B cells, and one of the B cells it will produce is for antigen A, specific to antigen A, and we will basically extract as many B cells as we can from the mice. We won't know which B cell is specific to antigen A yet. We're just extracting all the B cells that we can. And then we will fuse those B cells with myeloma cells because that will increase the shelf life, not really shelf life, but basically the life expectancy of these cells, of these B cells, because they don't survive well outside of the body. So they need to be fused with myeloma cells to make something known as your hybridomas, which have a longer life expectancy and a higher antibody production count. So we do this with all the B cells, regardless of whether they are specific to antigen A or not. Once we've done this step, we basically select for the hybridoma cell that is going to produce antibodies specific to antigen A, right? We select for it. Once we've selected for it, we isolate it, and then we use it to produce more antibodies, which is then collected and then administered to the patient themselves. Any questions here? All right. So far we've talked about dealing with cancer, dealing with tumor, but we don't really recognize our enemy. So let's understand the enemy a bit better. Cancer, right? So far, one of the most prominent things to kill humans on this planet and actually any living thing. Trees, trees get cancers too, right? Hope everyone knows that. Animals get cancers too, not just humans, but certain animals are better equipped to deal with them compared to others. In fact, whales, if I remember correctly, have some of the biggest tumors that you can ever think of, but they never have to worry about it their entire lives due to one of their biological mechanisms. If you have time, you can look into that. But we're looking at cancers in humans, right? So essentially, what is a cancer? It is a group of uncontrolled and unregulated cells that are replicating uncontrollably, and they basically start to take up parts of your body that they're not supposed to, right? Think of it like, let's say a cancer emerges somewhere on your on your kidney, right? Let's say there's a cancer emerging on your kidney. It will start to reproduce and grow uncontrollably, right? What started as a seed would grow and grow until it became the size of a grape and will continue to grow. But for it to be able to grow that much, it needs more resources. So what it does, it actually asks your body to supply it with more resources. So your body will actually make blood vessels and other systems to supply that tumor with more resources because it thinks your body is asking for it. Right? Your body thinks that tumor is a part of your body, so it doesn't question it, it just supplies the resources, and over time, that cancer grows and grows to become uh, danger unless we deal with it. So, what can actually bring about a cancer cell is basically when a mutations have accumulated in a cell's DNA, right? Our cells, not only do they have complex machinery, they've also been programmed to die, right? Apoptosis. Your cells will die when the time comes to it or when it receives the right signal from a different cell. So, for example, your helper killer cell, if you guys remember from your second line of defense, one of its main attacks is to tell a cell to kill itself, basically conduct apoptosis. This mechanism is what keeps certain infected cells, basically can deal with infected cells very early on. By telling that cell to kill itself, it will deal with all the pathogens inside it. But in this example with a cancer cell, what has happened is that mutations have occurred in the cancer in that cell's DNA such that this triggered death system has been deactivated, for one thing. Another thing is that it has been suddenly programmed to reproduce uncontrollably as well. So this provides a problem in that it bypasses normal regulatory checkpoints of a cell of the cell cycle and provide it with survival advantages. Basically, if there is the regular cell cycle where a cell is born, it does a job, it does whatever it needs to, and then eventually it goes to death on its own, or it is told to die by another cell, right? It's either it dies on its own or another cell comes up to it and it orders it to die. So that's the normal cycle of a cell. But in this example, so much mutations have accrued that it no longer follows that normal regulatory checkpoints of the cycle, and it will then just reproduce uncontrollably. So funny enough, your body actually is programmed to deal with cancer cells. It's just that it can't get them all, right? Your immune system is normally capable of recognizing cells that have developed mutations, right? All over your body, there are trillions of cells, there will be a lot of mutations going on. Then here, your immune system is actually programmed to deal with these cells that accrue mutations. But the problem is that your body deals with numerous cancer cells throughout that emerge. Not all of them can be dealt with. Some of them are able to actually develop mutations that allow them to even suppress the immune response against them, right? Your body's immune response can be suppressed by cells when they tell your immune system you're not supposed to you're not supposed to be acting up here, or you need to lower your activation right now because you're going to be a danger to the rest of your cells. If those cancer cells can do that, then they are basically circumventing your immune response. So this is something that can happen quite common. So on a daily basis, though, we actually deal with cancer cells that emerge in our body. We actually get cancer cells emerge in your body, your immune system deals with them. You never have to worry about it. But if you ever have a case of you're actually dealing with cancer, that means those cancer cells have been able to not just deal with your immune system, they've circumvented your immune system altogether. That's why immunotherapies need to be done where your immune system needs to be better equipped to deal with that cancer cell. So let's look at the monoclonal antibodies in relation to activation immunotherapies. So this is examples, like I said, where your immune system is improved to be better at recognizing and destroying cancer cells. And the key components can be used in a number of different ways. So two types of monoclonal antibodies used in immunotherapy. One is called naked monoclonal antibodies. The other one is called conjugated monoclonal antibodies. So naked and conjugal has to do with basically whether they carry any molecules attached to them. So for example, if you look at this picture here, this is an example of a naked monoclonal antibody because it's not carrying any other molecules, right? It's on its own. That's what it is, right? Whereas this one over here, it is a conjugated monoclonal antibody because it has a chemotherapy drug attached to it as well. So basically, anytime there's something attached to the antibody, it is a conjugated monoclonal antibody. If there's nothing else on it, it's just a naked monoclonal antibody. So we have three main mechanisms of action against cancer cells when it comes to monoclonal antibody, and each of them work slightly differently. Some of them don't even use antibodies. Some of them use your complement system. So let's look at it. The first one we're looking at is your antibody-dependent cell-mediated cytotoxicity. So how this works, you have your monoclonal antibody here. It binds to the antigen of your cancer cell, so your tumor cell or your cancer cell. And then once that binds to that antigen, once that antibody, monoclonal antibody binds to the antigen, it will interact with the cells of the immune system, giving priority to your natural killer cell, right? Your natural killer cell will use the monoclonal antibody, the fact that it recognizes the antigen, and then as a result, it will activate itself and actually start to target the cancer cell, right? So essentially, if you look at it here, monoclonal antibodies bind to the cancer cell and interact with the cells of the immune system, particularly your natural killer cells, causing them to get activated and recognize the antibody-coded cancer cells as a foreign cell and kill it. So these monoclonal antibodies basically make that cancer cell a target, and the natural killer cells will pick up on that and actually target the cancer cell, killing it in its tracks. Any questions about this one? Moving on. Next is your complement activation, right? This is where your monoclonal antibodies, again, will bind to cancer cells and interact with complement proteins. So the first one was it interacting with your second line defense cells, your natural killer cell. This one is interacting with your complement protein, your non-cellular component of your second line of defense. Make sure you remember what complement proteins are. In your second line of defense, there's cellular components and non-cellular components, basically things that are alive and things that are not alive, working together as part of your second line defense. The first, natural killer cells, they're part of the cellular. Complement proteins are part of the non-cellular components. So monoclonal antibodies work together with your complement proteins by basically identifying the cancer cell for the complement proteins. Once the complement proteins have been activated and they know that this is a cancer cell, they will destroy the cancer cell by forming a membrane attack complex, basically what we've looked at in the past, where complement proteins basically make a hole in the cancer cell. And as soon as you make a hole in a cell, obviously all the insides will flow outside, essentially killing the cell. So this is a way where it is activating your immune system. It is enhancing your immune system through complement activation. And the previous one is also an example of it, enhancing your immune system. Your monoclonal antibodies enhance your immune system to be able to target the cancer cells better. Any questions here? Monoclonal antibodies is. Whether this specifically, I can't tell. Moving on is your checkpoint inhibition. This disregard the picture, is just an example for you guys. This is basically when the immune checkpoints in your immune system are gonna be stimulated such that they will either allow the, let me find another way to say this. So immune checkpoints are regulators in your immune system, right? When they are activated, they suppress the immune system. That's why they are called checkpoints, right? They are basically making sure that your immune system isn't out of order. So this is to prevent an immune response from being so strong that it destroys healthy cells in the body. So some cancers can actually hijack the system and they secrete molecules that stimulate the immune checkpoints. And by doing so, they basically tell your immune system, bring it down. You don't need to act up right now. Cancer cells doing that to your immune system is a bad thing because they can basically quell the immune system from actually dealing with the cancer. Monoclonal antibodies then come back and block the immune checkpoints, meaning that the immune system is finally able to function at a greater capacity and destroy the cancer cells more easily. So if you have to think of it like this, let me simplify it. Immune checkpoints, they're your body's way to make sure your immune system doesn't cross the line, right? Doesn't overdo it. So cancer cells cancer cells hijack the system to tell your immune system to bring it down a notch. Monoclonal antibodies block that system, thus allowing the T cell to actually respond how it needs to destroy the tumor cell. All right, moving on. So with your monoclonal antibodies, there are also other ways that they can function, and they don't have to always be activating, enhancing your immune system or suppressing it. Some other ways is when you use conjugated monoclonal antibodies. These are basically antibodies with molecules attached to them, and they can be toxic to the cancer. So, for example, your chemotherapy drug, they can be actually attached to the antibody. And since these antibodies are going to actively look for the antigens present on the surface of the cancer, they are also specific to the cancer cell. That means that these chemotherapy drugs will actively attack only the cancer cell and not affect any other cell. Because remember, they are attached to the antibodies, and these antibodies will only bind to the antigens that are specific to that antibody. Okay? And then they will deliver those molecules to the cancer cells, killing them in the process. Any questions here? These drugs can have two main functions when it comes to the cancer cell. One is to either block cell growth by blocking the connection between the cancer cell and the proteins, or it triggers the cell membrane destruction or apoptosis. So it can either kill the cancer cell altogether, or it can prevent it from actually growing anymore. Any questions? All right. All right. Looking at monoclonal antibodies and autoimmune diseases, monoclonal antibodies can be used to suppress immunotherapy as well. This is basically when your immune system is being reduced or opposite of enhanced, lowered. Okay, so these reduce the immune system's attacks on cells that cause autoimmune disease, right? So what is an autoimmune disease? I hope you guys have heard of these before. This is basically when your own white blood cells fail to recognize your own cells and they end up inducing an immune response. So think of, let's say, let me just paint a very easy example. If you guys have heard of arthritis, a form of arthritis is when your own immune system attacks your joints such that the cartilage in your joints is no longer going to be there, and that can lead to early arthritis as well. So this is an example of an autoimmune disease where your immune system has gone haywire and isn't able to recognize self markers. Basically, the MHC class 1 markers that are found on our own cells. These lymphocytes have failed to recognize those, and they actually induce an immune response against those cells, right? So, and when this situation happens, that's when we have something known as an autoimmune disease, right? Your immune system is automatically attacking your own cells. Not really sure who came up with the name autoimmune, but that's how I think of it. The symptoms of an autoimmune disease are brought on by both B and T cells responding to self tissues as if they were foreign, right? Like I said, oh, that's actually a picture of that. So an example, right? Like I said, this is one of the reasons why you can get arthritis as well. It's a form where your immune system is no longer able to recognize the self markers that are found on your own cells, and they think since those immune cells can't recognize those self antigens, they will think those cells are actually foreign cells and trigger an immune response. And when that occurs, your body cells are basically being wiped out by your own immune system. Right? Okay. Moving on from this. So this is where suppression immunotherapies will act to actually deal with the issue, right? Because suppression is necessary because they will dampen your immune system or reduce its ability to attack. And this is especially important when your body is starting to attack your own cells out of no reason, right? It's just unable to recognize the markers on its own cells. As a result, your immune system is then now attacking your own cells. So we need to suppress our immune system by using suppression immunotherapies. Right. So thinking about suppression in immunotherapies, there are two specific that you need to definitely remember. Oh my God. Right. There are two that you guys need to remember. One is cytokine inhibition, and the other one is B cell and T cell depletion and inhibition. The first one, cytokine inhibition, that's when the cytokine molecules used in your immune system to coordinate its response are basically inhibited by the monoclonal antibodies such that it will actually reduce your immune response. So how does it work? Basically, so the monoclonal antibodies that are produced will actually bind to the cytokines, and thus they will not activate the immune response downstream immune response by the immune cell. So that's how cytokine inhibition works. It's basically, if cytokines are the messenger molecules between your immune cells, they trigger the immune response. Your antibodies will basically swoop in and take care of all those cytokines, and thus they will inhibit the immune response in general as well. All right, any questions here? Moving on is your B cell and T cell depletion and inhibition. This is when your monoclonal antibodies bind to autoreactive B and T cells and can be used to either inhibit these cells or stimulate other immune cells to destroy them. Basically, yeah, think of it like this, right? So it's either you target the cytokine before they reach the cell, or then if the cell has already been activated, you'd either target the cell itself. So if it's the cytokine you want to target, it's cytokine inhibition. Your antibodies, your monoclonal antibodies come in, they take up the cytokine, and then thus the immune cell is not activated. That's the quickest way, right? If they can't do that, they will then target the immune cell that's been activated, the autoreactive immune cell, where the monoclonal antibodies will bind together and hold the immune cell in place for it to be destroyed by another immune cell, or the antibodies will destroy the cell itself. So on more than antibodies by themselves do not kill cells. We are aware of that, yes. There are a few techniques that they use to make it an easier target for other cells, but antibodies by themselves do not kill other cells. There's only one part of your adaptive immune response that can actually kill, and that is your cytotoxic T cells, your T cell pathway, your cell-mediated immune response. That is the one that can actually kill cells. Your humoral pathway does not kill cells, they activate your antibodies. All right. So a majority of the autoimmune diseases that we have right now have no cure at this point of time. Instead, doctors are just trying to reduce the symptoms experienced by patients by using suppressive immunotherapies, basically suppressing a patient's whole immune system via immunosuppressive medication. So one example of when everyone would have to actually take immunosuppressant medication is right after surgery, right? We are asked to take immunosuppressant medication right after surgery because we don't want to get our immune system get triggered for stuff like organ transfers and stuff, especially early on, right after an organ donation, where you've actually given an organ to someone else. Their immune system can get very easily triggered due to the fact that it's a different organ from a different person. Even if you do have very similar blood types, you're very similar in a lot of ways, it's still a risk that that organ is recognized as being non-self. So the only way to help that progression, help your cells get more adapted, is to actually give you immunosuppressant medications for a while, let your body actually take in that new organ, accept it in, and then you can work on coming back from that. Whilst the use of immunotherapy to treat autoimmune conditions is a relatively new approach, it theoretically offers a lot of benefits. Immunosuppression would be far more specific, suppressing only autoreactive cells and leaving the rest of the immune system to function normally. There are currently a few immunotherapy options available for those with autoimmune diseases. However, most are still in conjunction with traditional approaches. So when we're dealing with autoimmune diseases, one key thing to notice is that you deal with this type of cell called a autoreactive immune cell. This is specific to autoimmune diseases, because autoimmune diseases is when your immune cells can no longer recognize the self antigens present on our own cells. So once they exist. We call them as autoreactive immune cells. These are the specific cells that cannot recognize the antibodies, the antigens on our own cells. So these suppression immunotherapies specifically target those autoreactive immune cells and leave the rest of your immune system intact. Any questions? Wait. Yeah, there's no questions. So summary, basically, immunotherapy is a form of medical treatment which modulates your immune response. So not really immune response, your immune system, such that it's better equipped to treat diseases or deal with diseases. It can either activate, enhance your immune system, or it can suppress or reduce your immune response. One or the other, depending on the disease we're dealing with. Okay, with cancer, we're going to activate it. With autoimmune diseases, we're definitely going to be suppressing it. So depending on the situation, we might have to do one or the other. It all depends on the type of disease we're dealing with. We also use monoclonal antibodies in the activation immunotherapy to help it recognize and kill cancer cells. And we've already looked at the three ways that it does that, right? Basically, we have your antibody dependent the ADCC, your complement activation, or your checkpoint inhibition. And with your your suppression immunotherapy, that is specific to your autoimmune diseases and how we can either do cytokine inhibition or B and T cell depletion and inhibition.